Okay, welcome. Thank you for joining us. I'm really pleased to say that today, as part of our Head Start webinar series, we are joined by my colleague, Professor Shireen Doak. Shireen is a Professor of Genotoxicology and Cancer here at Swansea University. And Shireen has kindly agreed to share with us her journey from her undergraduate degree with us at Swansea right through to her career and her role at the university now. So the title of today's session is Out of the Desert and Into the Sea. So those of you who are joining us live, if you do have questions, then you can pop them in the Q&A button at the bottom of your Zoom panel, and we will come to those at the end of the talk. And those of you who are watching this on record, if you do have questions following watching the webinar, then the email address to use to get in touch with us is study@swansea.ac.uk. Okay, without further ado, I'm going to hand over to my colleague, Professor Shireen Doak. Over to you, Shireen.
Thanks very much, Emma, and good afternoon to you all. So as Emma mentioned, I actually did my undergraduate in genetics here at Swansea University. So when I was asked to give this presentation just to look back at my career since my undergrad days, I realized with horror, actually, that I have been here for over 20 years. So I don't entirely know where the time has gone to, but clearly you can probably imagine I've enjoyed my time here, otherwise I may not have stayed for quite as long. So I started my undergraduate at Swansea here in 1997. As I say, it was in genetics, and I finished my undergrad in 2000. And like most students, when they reach the end of their undergraduate, they've got a big decision to make. What do you do next? And for me, there were two options. It was to go out in the big wide world and try and find myself a job, or to stay in research. And the reason the research side of things was really quite prominent in my mind at that time was because during the third year of my undergrad, I did a lab-based project with one of the research teams here at the university. And that project entailed me starting in the September period. And I actually spent 6 months with that research group doing quite a substantial laboratory-based project. And what that research project made me realize was just how much I really enjoyed the whole ethos around doing lab work and undertaking a research project where you had to try and generate new knowledge in an area that had been previously understudied. So in order to help my decision a little bit, I spoke to a few of my lecturers, and as it happened, one of my lecturers at that time, Professor Jim Parry, who's shown in the middle here, Jim actually had a PhD available. So we sat and talked for quite some time about what a PhD would entail. And really, after having that conversation with Jim, I was convinced that it was the next step for me. And as you can see from the title here, the PhD project itself was focused around trying to understand the molecular changes that happen in a condition called Barrett's esophagus. And Barrett's esophagus, this is an endoscopy image here that's taken with a camera down your throat. The entry here is the connection point, if you like, between the esophagus and the stomach. And patients with Barrett's develop this very angry red tissue in their esophagus just before the entry into the stomach. Now, whilst Barrett's is not a cancerous condition, it is pre-malignant. So a small proportion of patients with Barrett's go on to get cancer. And the problem is we can't always predict who those patients might be. So my PhD project was focused around trying to explore this tissue and understand if we could identify what we call biomarkers or molecular changes in either genes or proteins that could help us predict which patients developed a cancerous condition after having Barrett's. So this was a PhD that I was incredibly interested in. I applied and was very lucky to be awarded the position. And so I spent the next 3 years investigating just this question. I spent a lot of time in Morriston Hospital here at Swansea, collecting patient samples by working with the clinical team there, where they would insert these little brushes into the esophagus and collect cells for me, which I would then take back to the lab and undertake a whole range of analysis on. So some of the things that I would be looking for were changes in genes and chromosome numbers using fluorescent imaging technologies, such as the example image shown here. And then something else that I was also very heavily involved with was looking at protein levels in normal tissues as compared to both Barrett's and patients that had cancer, to try and see if we could detect those sorts of changes. So after 3 years of an incredible amount of time in the lab and writing a big book, essentially, my thesis, I was then examined, and in 2003 successfully passed my PhD. And so I was back at that gate again as to what to do next. Now, once you've done a PhD, you've normally got a few different options available to you. Some of my colleagues went into teaching, others went into working in industry, and others then remained in academic labs or private research institutes where they did a postdoctoral, where they undertook a postdoctoral role. And that's where you essentially continue working in a research program. And that was the route that I decided to take. It happened that Jim once again had a grant that had been successfully funded, and it was a position that he offered to me. So I very kindly accepted that role, and then spent another couple of years investigating a slightly different research question. So this time, what we were interested in was how exposure to chemicals that are in our environment, in consumer products that we utilize on a day-to-day basis, or in drugs, impact our DNA. And the reason that we were interested in this is that if you've got exposure to a substance that causes DNA damage, then that has the potential to cause cancer further down the line. And so understanding those chemicals and their safety is really very heavily regulated to make sure that we as a consumer population are not exposed to things that could cause us harm. And so what I spent time doing was investigating what was called a threshold model. So basically, at very low levels of exposure, our cells are actually very good at repairing their DNA. And so even if something did cause a low level of DNA damage, our cells can repair it. However, once you start getting to higher doses, you start introducing greater levels of damage, and all of a sudden your cells start to become overwhelmed. And this is what's known as a thresholded response. And so what we were able to do was try and identify where you've got safe exposure levels versus exposure levels that would cause concern. And so one of the methods that I used here was, again, fluorescence imaging based technologies to try and identify when we would see these fragments of DNA forming in cells because it was a result of DNA damage that had been induced following exposure. And this is actually a technique that, whilst I learned it back in 2003, is still a key technology that we use in many of our research projects today. So I spent a lot of time looking at quite a number of different chemical compounds and exploring these safe exposure level ranges versus levels that cause potential damage. And this was something that was of particular interest to a wide range of industries, because, as you can probably imagine, many industries are regularly developing new chemical compounds. And so they need to understand the safety implications of what they work on. And as a result, we had a lot of funding from companies such as this to continue work in this area. And I'm sure many of these names you may well recognize, but Unilever is a very big consumer industry. We then have AstraZeneca, which I think probably following the whole COVID situation is a company that everybody is very familiar with, given the Oxford AstraZeneca vaccine that's currently in circulation. And Hoffmann La Roche, which is another pharmaceuticals industry. So there was a lot of interest in this work, and they were really very generous in supporting further funding for us to continue this form of research at Swansea as well. Now, once I'd done 3 years of postdoc work, I did need to think about the longer term, because the one thing with postdoctoral work is that usually it's short-term contracts. And so at that point in time, I was starting to think about what I would need to do to have a bit more stability in terms of a more permanent contract. And whilst my options were still to go into industry, the other option was to look at a lectureship now and to stay in university setting. Now, to do that, one thing that I was very acutely aware of, thanks to Jim, because he was an excellent mentor, was that I needed to have teaching expertise and teaching skills, which at that point in time I didn't really have. I'd done a lot of lab work, written a lot of papers. And so in terms of my scientific CV, that was looking quite strong, but I didn't have much teaching expertise. And so in 2006, I decided to take a year out of research and focus on teaching. And what I did was secure a tutor's role in genetics here at Swansea University, where I spent a year doing nothing but teaching on the genetics course, large group teaching in terms of lectures, small group teaching in terms of tutorials. And so I really spent that time trying to get to grips with a whole academic side of teaching. But that still wasn't a permanent role. And so in 2007, there were RCUK Fellowships advertised for 4 positions at Swansea University that bridged medicine and engineering. Now, RCUK stands for Research Councils UK, and it's called something else these days, but at the time, they were very, very prestigious fellowships because they allowed junior researchers, such as myself at that particular time, to have a track into an academic lecturer's position. And it also provided you time to spend writing grants in order to secure funding to start to grow your own research group to become an independent scientist. So as I say, there were 4 of these fellowships available, but they were hugely competitive. So 4 of them were offered, 2 in medicine, 2 in engineering. They were in nanomedicine. So the idea was to try and foster links between medicine and engineering in order to really start developing quite what was at that time a very new field of research. So following an incredibly hefty interview process, there were 30 of us interviewed. I was very lucky to be one of the successful candidates based in medicine. And at the time, I'd spent a lot of effort really talking to colleagues in both physics and engineering, because they had available to them a whole host of imaging technologies, which they used for very high resolution imaging of materials. But I was looking at those imaging technologies and thinking we could actually use those for biological samples with a bit of work together to try and get the sample preparation side of things sorted out. And so that was really where our conversation started. And once I was awarded the fellowship, my research then started to diverge quite substantially, because I now had the freedom to really try and follow up on research questions that I had personally. And so this was a really exciting time for me. So it allowed me to do 2 things. One was to continue and expand on the cancer research side of things that I developed whilst I was a PhD student. But this time I'd moved into prostate cancer. And so the research that I started to kick off with the prostate cancer side was to look for molecular candidates that would help us to identify patients that were likely to develop aggressive prostate cancer, because it's something that's still quite difficult to do. And it does mean that some patients are overtreated. They're given fairly harsh therapies when perhaps they may never have actually developed aggressive disease. But that needs to be done at this stage because we can't predict which patients are going to get aggressive disease particularly well. The other thing is that by identifying biomarkers associated with aggressive disease, you now have new potential therapeutic targets that you can develop drugs against. And so this was another avenue of research that I was able to start following up, and I applied for grants which were very successfully awarded, and started having PhD students to work on this area under my supervision. But then I was also able to really expand some of the early discussions that I'd had with my colleagues in engineering and physics to see what we could do in terms of enhancing biological imaging using the technologies they had. And to give you an idea, this is the kind of imaging that we were doing at that time in medicine. So here what we have is a spread of chromosomes, and what we would do, for example, is label particular genes up with fluorescent tags so that we could pick them out to work out whether or not they'd been deleted or amplified in disease situations. However, once I started working with my colleagues in physics in particular, using this technique here, scanning near-field optical microscopy, we were able to get much more substantially higher resolution images. And what you can see here is actually one of these chromosomes at much, much higher resolution. So it opened up a whole new world in terms of analysis and technologies for analysis that we could apply to look for genetic changes. Following some of the conversations, though, with my colleagues in engineering, they realized that I had a background in safety assessment as well. And at the time, there was a lot of buzz about nanomaterials, nanoparticles, because these are incredibly small particles. They're less than 100 nanometers in size, and they've got really quite novel properties. And so this was at around about 2007, 2008, when my engineering colleagues were making these particles with a view to using them as a new material for a wide variety of different applications. But at the time as well, there was recognition that these particles might behave slightly differently in biological systems. And so they started to ask if I could look at the safety, because they were obviously producing these in the lab and handling them, and they wanted to make sure that their research staff were safe. And so I started to work with my colleagues then to look at the safety of particles that they were producing. And what we didn't realize was that this was going to become a huge area of research a few more years down the line. But this was very much an early starting point for us in 2008. And so from 2008 onwards, that was really when I started to become much more of an independent scientist. I was writing my own grant applications, which were successfully funded by places such as the Medical Research Council, but also several industries were really very interested in the work that I was proposing. And so that allowed me then to start growing my research group. I now had PhD students working on these projects, research staff, research technicians. And so the research side of things really did accelerate for me from 2008 onwards. And then in 2013 actually, I was approached and asked if I would consider applying for my professorship. And here we call it your personal chair. At the time, it was something that I was quite hesitant about because I was pregnant, but I was also relatively young as well to be applying for that kind of position. But I was encouraged to do so, and so I did. And 2004 was certainly a huge year for me, because in the January, my little man Riley was born. But then in the April, whilst I was on maternity leave, I actually found out that I was successful in being awarded my professorship. And really from there onwards, the research accelerated quite significantly. And in part, it goes back to that little story that I told you about with regards to the conversation I had around safety and nanoparticles with my colleagues, because what happened over the following years was that the nanotechnology industry really started to boom. At the moment, certainly by 2025, which isn't particularly far off, this industry is going to be worth just short of $175 billion. And the reason for this is that it is considered a key enabling technology. It's a really important industry that's underpinning a very wide range of different consumer products. The reason for this is that nanoparticles are very, very diverse. They're diverse in their size, in their shapes. You can have fibers as well as particles, right the way through to these really quite intricate structures here, which are called nanopepods. But the reason that these materials have gained so much interest is because they've got novel properties. They're incredibly catalytic, they're highly reactive, incredibly strong given their size, but because of their size, it allows miniaturization of everything that we use. And so as a result, this slide just gives you a bit of a feel for the number of industries that are already started to incorporate nanoparticles into the wide variety of products that they're developing. So for example, when you hear about self-cleaning windows, they're coated with titanium dioxide nanoparticles to allow for that. In terms of the aerospace industry, you've now got materials that are much stronger than steel, but much lighter. And so they're being incorporated and utilized in quite a wide range of areas. With respect to the medical healthcare industry, whilst they're not actively being used at this moment in time, they're being developed for a wide range of applications because these particles are so small, which means that they can be used to access compartments of the body that we may have struggled to do so with more traditional drugs. So whilst these materials are really exciting in terms of what we can do with them, there are, however, some concerns around their safety because they don't behave in the same way as chemicals. Now, for chemicals, we have a very well understood safety framework. And what's become increasingly evident over recent years is that because nanoparticles and nanomaterials are very different to chemicals, they don't actually behave in the same way in many of our safety tests as chemicals do. And so it means that our safety tests aren't wholly appropriate for evaluating these types of particles. We need new approaches. And as a result of this, there's been huge global efforts in terms of trying to better understand the safety of nanoparticles. And I was certainly in the right place at the right time for these sorts of questions to be arising because it was an area I'd been working on anyway. And so I started applying for major platform grants to address these questions. And they started from certainly 2016 onwards to be successfully awarded. And to give you a flavor, in 2016, I'm the co-director for the 14 million euro project CALIN, which is a joint project between Ireland and Wales. In 2018, became the coordinator of the 13 million euro project Patrols. And Patrols is specifically a nanosafety targeted project. So within Patrols, in the team that I've got 24 partners, and these partners are located in 13 different countries. So what we did was we had a very large research question to ask, and that was, can we develop safety tests that were specific to nanomaterials, but tests that allowed us to move away from having to evaluate safety in animals. So in other words, can we improve our non-animal tests on cultured cells by developing models, culture models, that better represented a number of different organs of the human body? Now, those sorts of questions a single research group can't address on their own. And this is why we had to have such a large collective of partners to really address what is a very, very big research question in a relatively short space of time of 3.5 years. So within Patrols, we've got partners that are mostly located in Europe, but we also have partners in Canada, the US, Japan, Korea. And so it's really allowed us to escalate our research to an international level. Following the successful award of both CALIN and Patrols, it then allowed us to start being invited into other large-scale research projects. And in 2019, we had 2 new projects that we're partners in or work package leads in, and it's Risk-On and Nanoinformatics. And then just this year again, we've become partners in another 2 multi-million-pound projects, Sunshine and Harmless. And all of these research projects are specifically driven at trying to improve our understanding of nanomaterial safety, develop new methods to allow for that, and also to develop new risk assessment frameworks so that we can more reliably govern what goes out into the commercial place and ensure the safety around those items as well. So this slide just gives you a bit of a flavour of the work that we specifically do at Swansea in these projects. As I say, one area that we're very, very keen on expanding is the development of more advanced culture systems that better represent the human skin, the lungs, the GI tract and the liver, because these are key organs of exposure when we either expose to something in the workplace, for example, you inhale them, in food substances that you may ingest, or in cosmetics that you place on your skin. And so much of our research is at the moment very heavily directed in this particular area of developing these more advanced culture systems so that we can reduce the need for having to test safety in animals. As I say, these kinds of research questions you can't deal with on your own. And this picture was taken when we were allowed to meet face to face with our project partners. It was actually taken just before the first lockdown in March of last year, where all of our partners in patrols had come together to discuss the science that we were all working on together. And so these sorts of meetings are really dynamic and exciting because you've got some of the best minds in the world coming together to really try and tease apart a challenging research question and come up with approaches and ways to better address them and to provide answers. And then this picture is our research group. So this picture, I would say, continuously changes as we have PhD students successfully completing, new PhD students starting, and the same with our research staff and technicians. But this is the in vitro toxicology group. So that's the name of our research group here in the medical school, and it's co-led by myself and then my colleagues, Professor Gareth Jenkins and Dr. Martin Clift. And we've got a really dynamic group, which are fantastic to work together with. And so just to wrap up, I wanted to go back to the original title of my slide, because it probably seems a little bit ambiguous given the talk that I have just given. So as I mentioned, I've been in Swansea now for over 20 years, but despite my accent, I didn't grow up in Wales at all. I grew up in Saudi Arabia. And I actually was there for the first 15 years of my life. I was there until I turned 15. I'd finished my GCSEs. And at that point in time, there weren't opportunities for women to continue education. So I came back to the UK in order to carry on to do my A-levels and to have the opportunity to go to university. And so that's really where the title of my presentation has come from, because my background is actually quite a mixed one. In terms of my schooling, I mean, I went to a British school out in Saudi, but it was an international school. So there was actually 63 different nationalities in school. And you could probably see this from this picture from one of my primary school classes. Looking back, I think we all took this experience for granted, but I certainly know now that this experience of growing up in that very diverse international environment is absolutely what's molded me into the person that I am today, and probably my attitude to life and what I do in terms of my day-to-day work. Because it's only through having a really diverse mixture of people that you can actually learn from each other. And that's an ethos that I absolutely continue in terms of my research. And hopefully, certainly over the last few slides, you can see how working together is absolutely vital in terms of really trying to move science forward. You can't do it in isolation. But working together is fantastic because you get to meet so many different people from so many backgrounds that have got very different viewpoints on how to do things. And it's only by having those different viewpoints come together that you can really start to make big step changes to move forward with. And so hopefully you can see just how working together is something that is factored throughout my education and my career to date. It's without doubt enabled me to really foster tremendous growth, both in terms of my own personal career development, but also in terms of the collaborative research ethos that I certainly strongly believe in. And it's something that I do spend a lot of time and effort trying to impart on the students that I teach as well, because I think that it's something that is important just in terms of career growth and personal growth more generally. And just in case you're wondering, whilst you've been looking at this picture, that was me. Okay, so that brings me to the end of my short talk. I'd just like to say thank you for listening, and I'd be happy to take any questions.
Fantastic. Thank you, Shireen. That was fascinating, and a lot that I didn't know. So considering we worked together for a number of years, so absolutely fascinating. So thank you so much. What so much you've achieved in 20 years that you've been with us as well. It's amazing to hear about. So those of you who are joining us watching this on record, thank you for watching. And as we said, if you've got any questions following watching this talk, then the email address to use is study@swansea.ac.uk, and that will come through to the right team, and we make sure we get those questions put forward to Shireen. So thank you for watching us on record.